Hi everyone, welcome to Science Talk. This is where we talk about topics and issues that are science related. And in this video, we'll be looking at the topic indicators in chemical analysis. As a scientist or as an analyst, you might be interested in knowing the status of the chemical substances or the reagents that you work with. You might want to know if they are basic, you might want to know if they are neutral, you might want to know if they are acidic. And it is very important as analysts for we to know when endpoints of a chemical reaction has been attained or when complete neutralization has been achieved in the case of acid-base titrations. So indicators play a vital role in chemical analysis. And in this video, we'll be looking at various classes of indicators, their applications, the various sources of these indicators, and the factors that could influence choice of indicators for chemical analysis. What are indicators? There are substances that exhibit visible change in color, pH, or some other physical properties when they come in contact with specific chemicals or conditions. Indicators are very useful in determining endpoints of a chemical reactions, and they can be used to detect the presence or absence of certain substances. And it is important to know that different indicators are suitable for different kinds of analysis. So their selections depends on several factors. You look at what are the requirements of this analysis or experiments that I want to carry out. What is the sensitivity of this experiment? So these are things that you need to consider before selecting these indicators. You don't just select indicators because you just want to use them. Are they suitable for such kind of experiment or analysis? Because there are certain indicators that are designed for certain kind of experiment. Classes of indicators. There are two major classes. We have the natural indicators and the synthetic indicators. So we'll start with the natural indicators. The natural indicators are the ones that are obtained from natural sources. For example, red cabbage is an example of a natural source of indicator. Turmeric is also an important natural source of indicators. Grape juice, curry powder, cherries, onions, and tomatoes are also important sources of natural indicators. But one important thing about these natural indicators is that they are environmentally friendly. So you don't need to be bothered of how do I dispose this kind of substances after use, because they are environmentally friendly. But they have some limitations because their color changes can be less distinct and their pH ranges may be limited when compared to synthetic indicators. Still looking at natural indicators and their sources and their behaviors in different media. Looking at the hydrangea flower is an important source of natural pH indicators. How does it behave in different media? In basic media, it gives a pink color. In acidic media, gives a blue color. Still looking at the chart on the screen, we have turmeric, which is an important source of curcumin. Curcumin is red in color when introduced into basic media, and then in acid medium, it gives a yellow color. And then red cabbage, which is a source of another natural pH indicator called the anthocyanin. The anthocyanin gives a light blue color in a basic medium, and then in acidic medium it gives a pink color, whereas in neutral solutions it appears and gives a purple color. And then litmus can be gotten from lichens and gives a dark blue color in basic media and then red color in acidic media. Still looking at natural indicators, litmus solutions naturally is purple in color, but in acidic solution it gives a red color, and in basic solution it gives a blue color. And then the litmus can also come in the form of strips of papers. We have the red litmus paper and we have the blue litmus paper. The blue litmus paper, when immersed into acidic solution, it gives a red color. And then the red litmus paper, when immersed into a basic solution, it gives a blue color. Turmeric solution, which is yellow in color, maintains its yellow color in acidic media and a deep red color in basic solution. The beetroot juice, which is purple, maintains its purple color in acidic solutions and gives a yellow color in basic media. And then the red cabbage juice, which is also purple, then giving a red to pink color depending on the pH in acidic media, and then green or yellow color in a base. The China rose solution, which is red, gives a magenta color in acids, and in base it gives a green color. Synthetic indicators. Synthetic indicators, these are indicators that are synthesized in the laboratory through the combination of various chemical substances at certain experimental or reaction conditions. Example of such kind of indicators are phenolphthalein and then the methyl orange indicators. And the choice between the synthetic and the natural indicators depends on certain factors such as specific applications. Maybe there are specific applications, and then what is the desired pH range for that kind of experiment that you intend to carry out, because some of these indicators have wider pH ranges while some have lower pH range. So the pH range is also an important factor to be considered when it comes to choice. And then how available are these indicators? Are they readily available? And the sensitivity required for such analysis, how sensitive are they? There are some indicators that are more sensitive than others, and there are some that are desired for certain kind of experiments. As we're going to look at these indicators and their applications, we'll know which one is suitable for which kind of experiment. Still looking at natural indicators. Now we have synthetic indicators. We have the universal indicator, which gives a green color. As you can see from my chart, we have various pH values and the various color that you obtain when you use a pH indicator. If you can see, at a pH of 0, the universal indicator will give a red color, but at a pH of 14 it gives a purple color. But at other pH it has different shades of color. If you notice, when it is in a basic media, you observe different shades of blue. Look at pH 8 is a light blue, pH 10 is blue, then the color begins to change and eventually changes into purple. And then looking at it in acidic media, also look at the various colors and the various shades of these colors too. And then phenolphthalein indicator, which is colorless at a neutral pH, and then also in acidic pH, also regardless of how acidic or less acidic or more acidic the solution is, it is still colorless in such kind of solution. And then in basic media, it gives various shades of pink. Methyl orange, which is yellow in color, at pH between 6 to 8, and then pH that is slightly below 6, it still gives that same yellow color. And then in a basic media, still maintaining that color, but at the pH drops down, going down to 4, 3, 2, you notice it begins to change gradually and eventually turns to red at this pH. The thymol blue, which gives a blue color at a pH of 14, and other pH between 10 and 12 and for 13, 14, it still gives those blue colors. But when you look at it at a pH between 6 to 4 and then let's say to 3, it gives a yellow color. But eventually at low pH between 0 to 2, it eventually gives a pink color. Bromothymol blue, it gives a blue color, different shades of blue in a basic media, and gives yellow colors in acidic media. Phenol red gives various shades of pink in basic media and gives yellow color in acidic media. Now let's look at the various types of indicators and their applications. We have the pH indicators. An example of such kind of indicators is phenolphthalein, which is used in acid-base titrations to determine endpoint of reactions. The bromothymol blue is used as an indicator in testing pH of solutions. Also, universal indicator, which is a mixture of several indicators that provide various or range of colors corresponding to different pH values, as we saw from the chart. Redox indicators. Example of redox indicators are potassium permanganate and methylene blue. Potassium permanganate is used as an oxidizing agent and also an indicator in redox titration. And methylene blue is also a redox indicator in analytical chemistry, and also it has biological applications also. Still looking at the various indicators and the applications, we have the complexometric indicators. Example of such type is the Aerocon black tea. It is used in complexometric titrations to detect the presence of calcium, magnesium, and other metal ions. One of the applications of this indicator is in the EDTA titration, where when you want to determine the hardness of a water sample using the EDA titrimetric method, then this indicator can find applications there, the Aerocon black tea. And then Calmagite is a dye used as an indicator to determine calcium and magnesium ions in samples. Precipitation indicators. Example is the silver nitrate, which is used to detect the presence of chloride ions in solutions. It actually forms white PPT of silver chloride. Fluorescent indicators. Example is the fluorescein. Fluorescein used as a fluorescent dye and also an indicator in biological and environmental analysis. Rhodamine B is also an example of a fluorescent indicator. It is used in various analytical processes such as microscopy and DNA analysis. We have the conductometric indicators. Example is tetraphenyl borate. It is used as an indicator electrode in potentiometric measurement, especially for determining cations. Chelating indicators is the last class of these indicators that we'll be looking at. An example is the EDTA, that's ethylene diamine tetraacetic acid, which is widely used as a chelating agent and also an indicator in complexometric titrations for determination of metal ions. So we have come to the end of today's training. Thank you for your time. You can subscribe to this channel to watch other videos that will be posted. So as we post new videos, you're going to receive them, you're going to view them, you're going to get notifications. Thank you for your time and thank you for listening. Have a nice day.